invite the next speaker, Dr. Parbati Biswas from University of Delhi, who will be speaking to us on misfolding and intrinsic disorder when a protein fails to fold.
So I thank the Academy to invite me to present my work, and today I am going to speak about misfolding and intrinsic disorder. So in both cases a protein fails to fold. So what is protein folding? It's a celebrated problem, and since proteins are direct products of genes, so it is important. Protein folding is important for the expression of genetic information in the cell, and in the schematic figure it is shown that how a protein is produced. So from DNA it transcribes to a single stranded mRNA, and by translation this mRNA is converted to a sequence of amino acid which folds to the protein. So this is the native state of the protein, but when the protein does not fold to the native state, it basically is misfolded. And the native state, actually the misfolded protein can fold to the native state through chaperones, or the misfolded state has, or the misfolded protein has options: either it aggregates or it is degraded to fragments and it is finally expelled out of the body. So now what is intrinsic disorder? Intrinsic disorder proteins, they basically do not fold. So that is encoded in the sequence itself. So for globular proteins which has well defined secondary and tertiary structures, intrinsically disordered proteins, the sequence itself encodes the disorder, and these intrinsically disordered proteins are very important because they are also found in human proteomes, and they have diverse function which includes the regulation of cell cycle control, signal transduction, molecular recognition and protein phosphorylation. So these intrinsically disordered proteins do not have well-defined secondary or tertiary structure, but they exist as dynamic ensembles of interconvertible conformations, which partially the proteins can be intrinsically disordered, which are called IDPRs or intrinsically disordered protein regions, and when a protein is fully disordered like this, then this is called an intrinsically disordered protein. So these intrinsically disordered proteins, they bind to suitable partners and then they can fold to a proper structure with well-defined secondary and tertiary structure. So intrinsically disordered proteins are also distinguished from the globular proteins in the charge hydrophobicity space by a linear boundary, where you can see that the native folded proteins, they occupy a region of high hydrophobicity and low mean net charge, whereas intrinsically disordered proteins, they have a preponderance of charged or polar residues and they lack a hydrophobic core. So they remain unfolded in natural conditions. So, so what happens? This protein folding can be explained by the energy landscape diagram. So where for the globular proteins, this is a funnel shaped rugged energy landscape, and the native state resides at the global energy minimum. So whereas for intrinsically disordered protein, we have a very shallow landscape with multiple minima, maxima and saddles. So this resembles the energy landscape of glass formers, and so here these intrinsically disordered proteins, basically they exist as interconvertible conformations, and for the globular protein, the energy landscape is rugged, but there is an overall bias towards the native state. So what happens for the why these disordered proteins or misfolded proteins are so important? Because they are implicated in many diseases. So like amyloid beta, so it causes Alzheimer's disease, and this many other intrinsically disordered proteins, they are implicated in neurodegenerative diseases, some forms of cancer, diabetes and etc. So you can see here the energy landscape for a aggregated or a disordered protein. So what happens here, basically all these aggregates which span from oligomers to fibrils, they are much more stable than the native state. So, once they are formed, they cannot go back to the native state. So, what we do here is that I will try to highlight some of the work. Of course, it is not possible to present all the work in this short time. So, what we do is that we try to distinguish the intrinsically disordered protein or the IDPs from the globular proteins by finding out the normalized relative composition. So, this gives the fraction of amino acids in the data set of the globular protein relative to that of the disordered proteins. And when we find the fraction of amino acids, there is a fuzzy boundary. You can see the red squares and the black dots are completely intermingled. But when we consider the amino acid triad, that is any amino acid and the flanking neighbors in the right and left side, we get a complete separation of the globular proteins, and the group 2 proteins represent the completely disordered proteins. So, this is the normalized relative composition. By the normalized relative composition of the amino acid triads, we can distinguish these two group of proteins. So, here also we find the entropy of, we continue with the study of these amino acid triads, and we see that though the disordered proteins lack a structure, so we can find out or we can estimate the conformational entropy of a disordered protein. So, whereas a globular protein, it approximately scans half of the phi psi angle space of the non-redundant database, whereas the disordered protein, it scans the entire phi psi angle space in the Ramachandran plot. Why? Because the globular protein has a preference for some structure, whether it is a helix or it is a sheet. So, it is confined to some region of the phi psi angle space, whereas disordered protein does not have preference for any structure. So, it can scan the entire phi psi angle space. And we try to explore this protein misfolding and disorder through protein design, which is basically an inverse protein folding problem where multiple sequences fold to a predetermined target structure. So, we design a theory for sequence ensemble based on some foldability criteria, which are generally energy based. And what is our contribution is that instead of assuming a single native state, we model the energy of the native state ensemble. So, we have a native state ensemble which has the native state as well as some of the energy of the low lying states which are close to the native state, and we also have the unfolded ensemble where the configurational entropy of the protein is very high. So, what we basically do is that we maximize the variational functional of sequence entropy, and we get a set of coupled nonlinear equations which we solve probabilistically, and we get the composition of a sequence in terms of site identity monomer probabilities and monomer pair probabilities. So, here we show that basically here we show that if you tune the variance in energy of the native state ensemble, you get 3 complete regions where the sequences are unfolded, they are folded and misfolded. So, in the misfolded state, the energy of any other state other than the native state, it is lower than the native state. So, here also we design some conformational ensemble with a definite similarity parameter, and we find that when this similarity parameter q is greater than a certain percentage, we get again these 3 distinct regions in the 3 distinct type of sequences. So, why does a protein misfold? Of course, this protein misfolds because of the cellular environment. It is determined by temperature, pH and mutation in the amino acid sequence. So, here we show that lactalbumin basically in acidic and basic pH, this represents a misfolded state or a molten globule, whereas in the neutral pH this retains its function. So, we also explored the role of point mutations. What are point mutations? Here we show that if we mutate a sequence, we get a mutated sequence, and in the mutated sequence basically we compute the clashing interactions and the matching interactions. So, this computation it depends upon the residue types and also where the residue is located, whether it is in surface or it is in the core. So, we show that the misfolded sequences they occupy a high clash and low match zone, whereas the folded sequences they have low clash and high matching interactions. So, matching interactions basically these are favorable interactions for amino acids and clashing interactions are unfavorable interactions. So, we also show that what is the role of local and non-local interactions in protein misfolding. So, we design this energy landscape all for real proteins. So, I have shown it for only one, but we have done for 4 or 5 proteins here. So, we show that the local interaction means the energetic interaction between a residue I and residues up to I plus minus 4. And the non-local interactions means the interactions between the one residue and greater than I plus 4 residues. So, here actually the non-local interactions determine misfolding, whereas the local interactions determine unfolding. And when you have the weight of these interactions are equal, that is weight of non-local interactions is equal to the local interactions, you get a folded protein. So, we also employ the same sequence design in disordered proteins with a different potential, with a coarse grain potential, where we take into account explicitly the hydrophobic interaction, electrostatic interaction, steric interactions, and we show that there is an order disorder transition at a critical beyond a critical mean hydrophobicity. So, basically this is the average Rg, which is a measure of the size of the protein, which is plotted against the mean hydrophobicity, and here we show that this inverted sigmoidal curve shows that there is an order disorder transition beyond a critical hydrophobicity, and this depends upon the mean net charge. So, higher the mean net charge, higher will be the critical, higher will be the mean hydrophobicity required for this transition. So, and what is the actual, how you measure this that the proteins aggregate really. So, for that we use reference interaction site model along with explicit molecular dynamic simulations. Now, one problem with the molecular dynamic simulations is that it is computationally too expensive. So, we resort to this kind of theory, which is an actually it is a statistical mechanical theory based on this Ornstein-Zernike equation, and this basically gives the total correlation function in terms of the direct correlation function and indirect correlation functions. So, we use some closure equations in the solvent density enhancement zone and in the density depletion zone to solve this, and basically we get the hydration free energy, which is a measure of the aggregation propensity of the protein. So, hydration free energy measures the affinity of a protein towards water. So, here it is shown like it is defined as the insertion of this protein from vacuum to a cavity. So, first this is the protein. So, we create a cavity here which has a free energy of cavity formation, then we insert the protein in the cavity and then fill it up with the solvent. So, there are several processes which measures this hydration free energy, and this hydration free energy here, what we do is that we take different disordered proteins and a globular protein ACP, and this MVFs 1CD3 and 1F0R have, these are partially disordered protein with different degrees of disorder, whereas synuclein is a completely disordered protein. And we show that there is a decrease in free energy with increase actually the free energy decreases more, it becomes more and more negative with an increase in the structural disorder. So, synuclein is a completely disordered protein, and here we decompose this hydration free energy into the electrostatic and non-electrostatic components, where we show that this hydration free energy of disordered proteins is mainly governed by the electrostatic component. So, we also do this for amyloid beta and its familial Alzheimer disease mutants. So, what we establish here is the partial molar volume. We use it as a thermodynamic order parameter to as a marker for its aggregation, as it's to measure its aggregation propensity. And we show that this partial molar volume, we have these causative mutants, they increase the aggregation propensity, and the protective mutants, they decrease the aggregation propensity. So, these are all natural point mutations in amyloid beta. And what we see that there is a change, this is basically negative for the protective mutants and positive for the causative mutants, and we correlate it with the experimental aggregation propensity, which is measured for amyloid beta in terms of the rates of these aggregation for the mutant as well as for the wild type, and we show that there is a correlation which exists. And we do the same thing, the change in hydration free energy. So, this is primarily negative for the protective mutant and positive for all these causative mutants. So, which shows that there is also a correlation with the aggregation propensity here. So, you can see that more positive hydration free energy, that there is more aggregation propensity of the mutant. And we calculate all these things also, and we show the effect of charged residues on hydration free energy, where when the protein has net positive charge, then positively charged residues contribute more than that. And we also study the kinetics of protein misfolding through master equation, and we solve it to give the MFPT, which is the mean first passage time. This is the average time required for the protein to reach the misfolded or the aggregated state. So, these are my conclusions, and I thank all my students. The face of lab has changed, and I thank all of them for their contributions, and I thank SERB and CDAC BRAF for allowing me to the computation time, free computation time, I should say. Thank you.
Let's have a couple of questions.
Basically, the shape and minima distribution of the energy surface, because in the Ramachandran plot you showed, there was single cluster in one case.
That is for the.
It was just two clusters.
That is for.
This
is a schematic diagram. It is not the exact result. And schematically, I tried to show that if you have amino acid triad for the globular proteins, these are restricted to only some portions of the Ramachandran plot, because they have either the propensity of helix or sheet. So, they do not populate the entire region of the dihedral angle space, whereas the disordered proteins, they do not have any structure. They are like random coils, these are non-compact coils. So, they populate the entire region of the Ramachandran space. And in this, we show that even without knowing the structure, we can get the conformational entropy. You have to just sum the conformational entropies of the amino acid triads. Yes.
Thank you.
We'll have two more questions. Ramsesh first, Dr. Ramsesh.
Hi, I was just wondering what kind of an order parameter you have for this to characterize the disorder. Do you have any kind of an order parameter that you can
Actually, we do it with the RG. So, there is a sudden jump in RG, you will find the inverted sigmoidal beyond a certain hydrophobicity, below a hydrophobicity cutoff, there is a sudden jump in the size of the protein. So, basically disordered proteins have a larger Rg and globular proteins have a smaller Rg. So, this we show it for different mean net charges. Of course, the charge hydrophobicity boundary is very important for disordered proteins.
So, nice presentation, ma'am. So, what are the factors that determine intrinsically disordered protein as a stable as compared to the this globular protein? Because some of the cases you shown they are
Intrinsically disordered proteins can bind to suitable partners, be it ligand, be it other proteins, be it DNA, and then they have a proper structure. Otherwise, they are stable, but they have very shallow minima, as I showed the energy landscape, so they can switch between the conformations.
Okay, thank
And
you.
the barrier is very, very low. Whereas the globular protein has a deeply funneled energy landscape, though the local barriers near the native state where the protein gets misfolded, they are very low. But the barrier between the folded and unfolded is very high. The unfolded has a much higher configurational entropy and the native state has a very low configurational entropy.
Thank you.
Let's all thank Dr. Parbati Biswas for a wonderful presentation.